extra long presentation.
All right, thanks a lot. I'm a junior fellow at the Roland Institute, and these are the past and present members of my lab. What we're interested in answering is, or addressing is, how does a limb or how does a muscle control the motion of a limb? And from a muscle physiologist perspective, we're interested in what governs muscle power output. And if we understand something about that, then we can understand maybe what limits how fast an animal can move during locomotion. Our approach to this is to build a muscle-powered swimming robot and stimulate the muscle over a range of conditions. I'm going to be talking about swimming, but the methods are also interesting potentially for future work in terrestrial locomotion. So I choose swimming as a mode of locomotion because I view it as a fairly straightforward problem. The task is just to move forward. And I choose frogs as a model system because a lot is known about their muscles, and also they're very diverse. So that's kind of like a natural solution space to explore. The specific frog that I'm interested in looking at for now is Xenopus. This is a purely aquatic species, and as you can see, it swims by rotating its feet, and that rotation is powered by the calf muscle, the plantaris. And for a long time, I've been thinking about things from the muscle's point of view, but more recently, I've been trying to think more about what's happening about the complex ways that limb is loaded while it's swimming. Now, robotics are very useful tools for understanding the external forces on limbs, but maybe a limitation is that although we would understand those external forces, we may not understand the underlying internal forces from the muscles required to move those limbs. So one way to address that is to do an in vivo experiment where you measure the strain patterns of a real limb, then you replay those length change patterns into a servo motor that oscillates this in vitro isolated muscle while you stimulate it and you get muscle force out, and you can record muscle force, work, and power. So this is a very powerful technique, but that also has limits. So Marsh pointed out that without knowing the in vivo situation, we don't know necessarily which conditions to test, which strain patterns to test. And the implications of this is that if we want to address a simple question like, for example, would frogs swim faster if they had a larger foot? We couldn't address that question with this technique because we wouldn't know, that would be load dependent. We wouldn't know how this muscle strains. And just to reinforce that, I think about a muscle first as isolated in space. So it has a force-velocity relationship and a force-length relationship such that its power can be written as a function of length and velocity. And so the muscle has to operate either on or below this surface. So that's kind of the performance space of the muscle, theoretically. But when you attach a load to this muscle, my intuition says that this performance space is constrained. And so the entire research program in my lab focuses on how the load influences the behavior of muscle. And this work, the methods of this work has been inspired by a recent work in Hugh Herr's group down the street. So one technique we use, we call the in vitro robotic method, is where we have this muscle attached to an ergometer, we stimulate it, we measure the force is sent through a real-time controller as a command signal to a robotic foot, and that applied torque causes the robotic foot to displace. That displacement information is sent back to a linear length change, close the loop, causing the muscle to shorten as if it were directly attached to the robotic limb. So effectively, if I take my hand and move this part back and forth, I'm actuating the motion of the robotic foot. But I'm also feeling the force of that foot as it's sloshing against the water. So what we can do then is we can change the foot size or shape or material properties, or we can change these software coefficients that deal with the displacements and the forces to change the muscle moment arm, so virtually changing the skeletal structure to look at gearing effects. So we have that, but we also have a complementary approach where we call this an in silico robotic method, where we just delete the real muscle, keep the robot, and so we have our angular change coming in here, and then instead going to a hill-type muscle model, much simpler than the previous talk, to get our force for the next time step. So this is a complementary method, and it's activated by this activation model. And just recently, within the past few weeks, I've gotten to work a tendon component in here, and we can control the stiffness of the tendon in software. And I have to thank Greg Sawicki for helping me conceptualize this months ago. So what this looks like, here I'm going to be, whoops, if I can get it put back, okay. So I'm playing around with this lever here with my finger. The orange trace is muscle force. So as I'm wiggling that back and forth, I can feel the water force sloshing back and forth on the fin. You can see the fin rotating. The blue up and down is hydrodynamic force sensed by a separate sensor at the base of the fin. So we can also make this thing swim forward like a frog. So we take that hydrodynamic force, and that goes into a separate feedback loop, a separate control loop. That hydrodynamic force now becomes the thrust, goes into a mathematical model that says if there's an imaginary body with a mass and a coefficient of drag and an added mass coefficient, then the acceleration on that body should be this. We can integrate that in real time and get a target velocity for this forward motion. So the swimming behavior emerges as the muscle pulls on the lever. What this looks like physically during an experiment, this is just looping back and forth. It's hard to see, but the muscle is shortening here, and you see the fin moving through. This is not water, this is like some crazy goop. But anyway, that's what it looks like in real life. The cleaned up video looks like this. So the muscle is rotating, or the muscle is contracting, and the fin is rotating in response, and then swimming forward. Here we have the only input to the experiment, the stimulation spikes to the in vitro muscle, the muscle force, the velocity of the muscle, and the hydrodynamic force. So the question that I'm going to address today is, can the morphology of the limb be tuned to increase muscle power during swimming? So the morphology I care about is, for example, this oar blade that rotates around the joint, and hydrodynamic force appears at some distance, the out lever, and then we have the in lever distance on the other side of the joint. The ratio of the two is gear ratio. So a high gear ratio, we'd expect very small muscle displacements for a large angular excursion. So maybe I'd expect a really powerful swimmer like Xenopus. The plantaris muscle would be in a high gear ratio so that it can generate a lot of force against the water. So that's what I would expect. So I tested that, increasing the gear ratio by twofold each time. So you can see that the muscle force increases and the velocity decreases dramatically. And you can also see that the force and the velocity are in phase, which makes sense because the muscle is loaded by the hydrodynamic drag force, which is a function of velocity squared. So we can take this whole thing out of the water and do inertial loading, and I kind of think of this as the swing phase because limbs in water, they can collapse, and so then inertial forces become important. So here we see that force is out of phase with velocity because, of course, the muscle is loaded by the acceleration of the limb, or the inertia of the limb. So what's interesting to me is looking at the power output. So when the muscle is against a hydrodynamic load, as we increase the gear ratio, power goes down against what was expected, but power goes up in air. So what's going on here? In order to get some perspective, I plotted muscle shortening velocity, average shortening velocity against power. Triangles are fluid loading, and then circles are inertial loading, for low, medium, and high gear. And so for perspective, I put the Xenopus levis power velocity curve that we just recently measured up on here. And so you see this point is near maximum power. And so we see that for inertial loading, we're starting at a relatively high velocity and kind of marching over this peak. And the starting point is different for a hydrodynamic load, but the principle is the same. We're kind of working our way through velocity power space. So that's useful to know, and one thing I really wanted to know is what governs how these points, what governs the relationship between power and gear ratio. Long story short, you have to do a model to get at that, and you plot muscle force velocity relationship against the increasing force velocity relationship of fluid. So it's a velocity squared relationship. Long story short, where these two curves intersect dictates the power of the muscle, the power output of the muscle. So this is a case where the load is determining the operational regime of the muscle. And so we can make kind of a hypothetical, a theoretical power map. So based on the morphology of the limb, so a small muscle or a large muscle, small foot, just the ratio of the two, can dictate the optimal gear ratio for swimming. And it turns out that it seems like strong swimmers in nature are seeming to be towards the optimal end of this space. So anyway, just to conclude, I'll just show some more current stuff. This is we're working on internal elastic structures, looking at muscle force and hydrodynamic force. We're also looking at external elastic forces. And I'll end there, and just acknowledgements. Is that a good place to end? Yes.
It wasn't clear to me, is all your analysis done over a single sort of kicking stroke?
Yeah, it's just one stroke for now. It's just basically a power stroke. So the behavior that I'm emulating is when you scare a frog, usually a lot of times there's just one kick, glide, and then they stop. Sometimes they do repeated strokes. I do want to get into repeated strokes, but I just don't have the physical translational distance right now, and that's under construction at the moment.
I was curious where you put that pressure receptor on the frog foot for the sensory feedback into muscle.
It's not feeding back into the muscle. So are you saying the hydrodynamic force sensor bit of it?
Yeah, well, I'm wondering where it was placed on the foot, because would that affect how the software is going to read what pressures?
Exactly. Yeah, it's not a pressure sensor, it's actually a force sensor. And that's a great point, because you're exactly right. So the cool thing about, a colleague of mine designed this sensor, and here are some strain gauges here. And so basically, it measures the force independent of where that force is acting. So I don't know if that answers your question, but that's a great point. Yes, sir?
So I'm a little concerned that if you had studied bumblebees or fish, you would have somehow missed the mechanical resonances that really allow them to get power output. In insect flight, it's not just the muscles, it's the whole mechanical structure.
Right.
And maybe a frog resonates in some way, or hydrodynamically resonates in some way.
Yeah, I don't think the analogy holds. That case with insect flight is a special case. And that's when the cycling of calcium is relatively slow, so they're getting around that by. So these contractions aren't cyclical. So if frogs had to get to where they wanted to go by oscillating their limbs really fast, then we would have to start thinking about the resonance of the system. But I don't think that applies in this case. This is basically a one-shot kind of ballistic kind of thing. I don't know if that answers your point.
So are you saying that they thrust and then glide for a while?
Yeah.
And then, so it's not, so they're not just repeat the cycle?
Exactly. It's not a cyclic behavior. Yeah. But if it were, yeah, I mean, you're correct. If I was modeling a cyclic behavior, I would have to account for the mechanical resonance of the system. Yeah.
So half question. Could you explain the rationale for so you have these setups where there's the muscle, there's the software, and then there's hardware as well. It feels like you could study the muscle alone, or study the hydrodynamics alone, all of which will be simpler problems, and you can understand it. So why put them together and study how?
Because if you don't put them together, you don't get the right answer, in my opinion. So here's a case where we've changed the stiffness of the fin, And so not only does that, that changes the hydrodynamic force, and it also changes the underlying muscle force, and therefore it's going to change the motion. So since it's a coupled system, you can't mess around with one parameter and ignore the other parameters. I don't know if that makes sense. So basically, it's a coupled system, so the motion output is going to depend on the forces, and the forces are depending on that motion. And then if you have some flexible fin involved, then that's going to influence things as well.